Okay, this is a quick review for Lab Practical 2. There are a couple additional things that are going to be on this short talk that will that I recommend that you'd study or go over that are included also in this slide. So anyhow, first of all, this is kind of has to do with I believe it was lab 6 or lab 7, and it's about the phyla belonging to the human microbiota. And it was, actually, it was Lab 6. We worked with the firmicutes and the actinobacteria. And these are some of the feature of those two phyla. The firmicutes, of course, are gram positive. They also, their shape is usually cocci or bacilli. And again, the firmicutes have those low guanine and cytosine ratios. The actinobacteria is another phyla that is gram positive, and it has a gram positive cell wall. The shapes in this phyla are some cocci and some bacilli, but many of them are odd shaped. They're filamentous or they may be branching. These are the guys that have the high guanine and cytosine ratios. Now, the acid fast bacteria belong in the actinobacteria, and of course, an example of an acid fast would be your Mycobacterium tuberculosis. Now, at the top there in the blue bubble, the proteobacteria, those are the bacteria we worked with in Lab 7. And those are the guys that have gram negative cell walls. They have a variety of shapes, as you see: bacilla, vibrios, spirilla, cocci, and also filamentous. So they are very diversified phyla. The Bacteroidetes we didn't work with. They are difficult to culture in a standard laboratory, but these are gram negatives also, and they are motile, most of them, and also bacilli. So the takeaway from this is I would suggest that you know which phyla are your gram positives, and that would be your firmicutes and your actinobacteria. Also, the common shapes in each of those phyla. And again, I talked about this in lecture. The low guanine and cytosine bacteria belong to the firmicutes, and then the high guanine and cytosine belong to that other phyla, that actinobacteria. The proteobacteria, again, these are the groups, the gram negatives in Lab 7, and these are the group of bacteria that we did the IMVIC test on, the proteobacteria. And there are 5 subgroups to that. We specifically worked with a gamma group or subgroup of the proteobacteria. All right, now on the gamma proteobacteria, these terms were mentioned several times. The glycolytic facultative anaerobes, and these are guys that are facultative anaerobes, and they use glucose as their food source. They are catalase positive but oxidase negative. Okay, catalase positive, oxidase negative. Now they grow on TSI, but both on the slant and the butt. So this means they can grow aerobically and anaerobically. And the big thing to remember about these glycolytic facultative anaerobes is that they can ferment. They undergo fermentation of glucose and or lactose and sucrose. Okay, so glycolytic facultative anaerobes. The pseudomonads are aerobes, usually strict aerobes. You look at the catalase and oxidase test, and they both will be positive. So this is telling you that it is a strict aerobe, but they belong to the subgroup Gamma proteobacteria pseudomonads. Okay. They will grow on the TSA slant, but not on the butt. and there is no fermentation with these guys. So again, these two terms are used in Lab 7, and I think it's referring to the gamma proteobacteria, those gram-negative bacteria. It's just telling you basically um, what the test results are and how they live, okay, and what they ferment, or if they even ferment at all. So I would suggest that you kind of review um, these terms. Now this, you may have some of this, you will have some of this on the lab practical. You remember the oxidase and catalase test. We aren't going to be asking so much what enzymes they are, um, what's going on in them, but we'd like you to remember, oxidase positive, catalase positive, those results, what kind of oxygen requirement would that bacteria need? So in the case in the previous slide, I said if it's oxidase positive, catalase positive, and the TSA maybe has growth on the slant, but no fermentation, no yellow development, you can say that is a strict aerobe, okay? If the catalase is positive, the oxidase negative, and then maybe on the TSI you may have some fermentation, and if there's growth on the surface and growth in the bud of that TSI, realize again that that's a facultative anaerobe. So I would say if you need a little review on that, go over the lab practical one quick review, or your previous lab, it may be lab 4. So there may be a couple questions on that on the lab practical. Now I'm going to go over again the selective and differential media, and we use this media, or the 3 types of media, for the food lab. And a couple of you may have not made the food lab, so this will be a really good review for you. But we worked with MacConkey's, sorbitol in MacConkey's, and also the mannitol salt agar. In the MacConkey's agar, again, kind of go over what the selective ingredients are. That I think is your biosalts and crystal violets. But I want you, you should just go over that, what's in these medias, but also after incubation, what happens, what does it mean? And in this case, if you've got the MacConkey agar and you've subbed it with bacteria, if there's growth, it's a gram negative. Okay, only gram negatives grow on the MacConkey, and usually it is a gram negative bacilli. You're also looking for color change. If it's pink, know that means lactose, which is in the MacConkey, has been fermented by those gram negative bacilli. Okay, and that means they turn pink if there is fermentation. Now normally we don't work up too much of a sweat with that, because a lot of times if it turns pink, if there's fermentation of lactose, it is a gram negative bacilli, but it does not cause, there's not a high risk of acquiring foodborne illness. If there is no color change with your MacConkeys, this is what we want you to realize. That means what? That lactose is not being fermented. It is a gram negative bacilli because there is growth, but that lactose is not being fermented. And these may be associated with foodborne diseases, some serious foodborne diseases by the 2 bacteria listed here. Salmonella, which is a gram negative proteobacteria, or Shigella, another gram negative proteobacteria. And if you look at these on the MacConkey, they will have no color change. So how do you determine which genus that bacteria belongs to on that MacConkey if there is no color change? So that you do these follow-up tests. And the follow-up test would either be a TSI which you all are familiar with, or the SIM, which you are newly familiar with. So in the case of the Salmonella, it would have a red over yellow butt and be black because it's producing hydrogen sulfide. It undergoes anaerobic respiration, a type of anaerobic respiration. Now, for the SIM, the Salmonella will be positive, excuse me, for hydrogen sulfide production. So it will be black, the tube will be black. And Indo will be negative, okay? Indo is not produced. And then it will be motile, so it'll be a cloudiness. The bacteria itself is motile, so it will be cloudy in the agar. Shigella, I've got the TSI there, is going to be red over yellow. No hydrogen sulfide is produced. The SIM, it will be negative for hydrogen sulfide production, the S. It will be variable as far as indole. I guess it depends upon the species of Shigella. But the M for motility, they are non-modal. So if you would have to determine whether it's a Salmonella or Shigella, you do the follow-up test. Just realize that the Salmonella, the big thing with that is that it produces hydrogen sulfide. It will be black in the TSI tube and also black in the SIM tube. Okay? And another difference is Salmonella, the bacteria, is motile. It moves, it swims. And Shigella, it does not. They're non-motile. Okay, the next one is your MacConkey sorbitol agar. So again, as a nutrient, instead of lactose, you have sorbitol, which is a sugar alcohol, in the agar as their food. Now, most strains of E. coli will ferment sorbitol. And again, what's the result? You get your pink or red colonies on the SMAC agar. So you see these red lines are pointing to your probably E. coli. They're fermenting the sorbitol and they've turned pink. Now, what do we worry about with MacConkey's sorbitol agar is if there is no color change. And if there is no color change in the colonies, and that's what the gray arrows are pointing to, this is kind of a mixed culture in that it's different strains of E. coli. If there is no pink color, no sorbitol has been fermented, it means it's the pathogenic E. coli 0157H7. Okay, that is a hemorrhagic strain of E. coli and a deadly, can be a deadly strain. So I guess with the MacConkey's and the MacConkey sorbitol agar, no color change means it's going to be one of the foodborne pathogens. Okay. It just depends on the follow-up test and which type of agar you are using. Okay. Now, mannitol salt, you all are pretty good at knowing this. Again, I just want to tell you that those bacteria that grow on it mean that they are salt tolerant or halotolerant. They use a 7.5% salt solution is mixed in with this agar. So if there's growth on there, they are salt tolerant. Now, we kind of think that Staphylococcus is the only genus that can grow on salt agar or MSA, but there are some other species here, Micrococcus, some Bacillus, and also Enterococcus. But for the food lab, and usually when we're talking about food poisoning, and we use mannitol salt, it's for the confirmation of species, of differentiating species of Staphylococcus. So in this case, we are looking for fermentation again. And in the case of the mannitol salt, if there's fermentation, the bacteria will create an area of yellow around the colonies. So in this case, if you look at that mannitol salt agar, yes, you have A and C have bacteria growing on them. B, there's no growth, so B does not tolerate salt. A and C can grow, are halotolerant, but only A ferments mannitol. Now, so what ferments mannitol are your Staph aureus. So you'll get a yellow, a yellow color with the Staph aureus, but there are some other Staphylococcus. So you have to further test that mannitol fermenting bacteria to confirm that it is Staph aureus, and that is done with the coagulase test. Okay, that quick card test. And if it is Staphylococcus aureus, you will get a strong clumping because it clots blood, and it will kind of clot the glass coated fibrin beads on the coagulase test. So in other words, in order to confirm that it's a Staph aureus, if you get yellow on the plate, the confirmation test would be your coagulase test. And then this is another, the IMVICs, okay, the IMVICs, and these are done on the proteobacteria to differentiate among the species of them. And know what these stand for. I have that in the review, but I'm just going over it again. The I actually has, we're checking for indole, but along with that, it's a 3-for-1. It's set up in the SIM tube. You are checking for S, hydrogen sulfide production, and M, motility. So it's a 3-for-1 in that I, with that I. Hydrogen sulfide production, indole, and motility. Methyl red is the M, and Voges Proskauer, named after two gentlemen, Voges and Proskauer, is the V. We used the same broth, if you remember, the MRVP broth, into each of those tubes and then inoculated them with our bacteria. And after incubation, you will notice after adding methyl red reagent, if it's positive, it will turn red. Voges Proskauer, you add VP1 and VP2, two reagents, and if that turns red, it's a positive Voges Proskauer. And what these both are basically kind of different types of fermentation. You start out with glucose, but again, I was telling you there's different pathways of fermentation, okay? In methyl red, you get mixed acid fermentation, so mixed acids are the products. In your Voges Proskauer, you're getting not so much acidic products, but alcohols and other products. And finally, in the IMVIC, the last letter is C. That's for citrate utilization. It's that green plate there. So it has a different indicator, as I talked to you in lab, but also what it has is citrate in it. And so these bacteria, if they're positive for citrate utilization, or they use a citrate, it will be, that citrate will then, once it's broken down a little, will be incorporated into the Krebs cycle. So it kind of bypasses the first step of aerobic respiration, okay? And it will turn blue if it's positive. So make sure you know what each of these tests are, or the basic steps in each step, and what IMVIC stands for. And these are done as a differentiating test among the gamma proteobacteria, or the bacteria that belong to the Enterobacteriaceae family. I keep saying that. All right, the last thing, you're going to have some epidemiology problems. So please please know how to calculate these equations. If you're asked for incident rate, this is what you do. Prevalence rate and the case fatality rate is the number of people who die from an illness divided by the number of people confirmed to have the illness. And in this case, if you see in a problem number of people confirmed to have illness who have been hospitalized, that number goes on the bottom of this equation, okay? And that will give you the case fatality rate. Finally, on the risk ratio, these are these word problems. There may be one or two of them. Be able to calculate the risk ratio. And again, that's the one that's done in table form. And it's actually just the incidence of the outcome of an exposed group divided by the incidence of the outcome of the unexposed group, or plug it in using that nice little formula down there. Many of you students seem to be able to do that. If you have any problems with understanding any of this, please, I will have an office hour Monday night from 7 to 8. You can ask me then. I also, for the Thursday group, will have my office hour from Wednesday, 12 noon to 1. And I wish you luck. I hope this has helped a little bit. Again, just study. Practice some of the problems in the lab, or re-practice them, especially for epidemiology. And good luck. I will see you either Tuesday or Thursday. Good night.